excited to move on into this technical portion. Again, I don't want to lose you all in the weeds. So remember, this is about your creative freedom. And, I've asked students in the past, who knows what seed saving is? And they've just, laid it out pretty clear to me. It's like, seed saving, really? Is that just the process of saving seeds? And I'm like, okay, I don't know why I hadn't thought about that before. I guess you're right. Yeah. So yeah, in the end, I just want to let you know that while this may seem like there are a bit of rules or processes or steps involved, in the end, saving seeds is the same as seed saving, and seed saving is as simple as saving seeds. So yeah, don't get all worried about it, and experience will really provide you with that holistic knowledge. I just wanted to say that. Sorry. Thank you all again. Okay, so let's jump into it. Now, moving into this second step of the seed cycle, let's discuss the processes involved to clean, dry, and store seed. Basic seed saving supplies consist of household or classroom items that typically can be located and gathered with ease. If you do not currently possess some of these tools that you see on the slide, I really encourage you to think creative and improvise with other tools that can perform similar tasks before purchasing anything. I have found it helpful to have a wide assortment of these materials kind of compiled into a seed saving kit-like setup so that when the time comes and your seeds are mature, you can harvest, process, and store and save anything that comes your way with no delay. So we have just harvested our seed, right? Now at our processing station, we need to make sure that every seed variety has a label that can follow it from cleaning to drying and eventually storage. While labeling may seem like a small step, I would argue it's actually one of the most important ones, especially if you're growing multiple varieties of a similar looking seed. Imagine if you're growing 2 different tomato varieties. I mean, it's pretty hard to tell a tomato variety apart by its seed, unless you have the tomatoes in front of you. So don't forget to label your plants. Include the crop type, the original source of the seed, the date and location of harvested, of course, the variety name, and other relevant notes about your seed saving experience. Before we dive in to the different classifications of seed, we all need to have and ensure that we are saving and selecting for healthy seeds. We know that healthy seeds lead to healthy plants, and healthy seeds or healthy plants lead to healthy seeds. So again, before we begin saving seeds, we need to know what a healthy seed looks like. There are 2 main things that make a seed healthy. One is that they are fully formed, and the other is that they are viable and alive, meaning that have the potential to germinate and grow into healthy plants, of course. If seeds are not viable, they will not even germinate. If a seed looks large and plump, it is more likely to be viable and filled. A healthy seed has an embryo and endosperm taking up space on the inside. And in contrast, a seed that looks shriveled up or malformed is actually more likely to be empty or inviable. Additionally, if you are, seeing any mold or holes in your seed collection, it is likely the seed has been infected by fungus or insects. When saving seeds, seed selection is the necessary process of removing seeds that obtain such attributes. Again, holes, fungus, it's malformed, looks shriveled. You'll remove seeds that have those characteristics just to ensure that your seed lot is not contaminated during storage and to ensure that you have the best quality seed. The particular procedures involved when saving seed are largely based on the principles of seed biomechanics, which categorizes seed not on the basis of biological, classifications such as natives or non-natives, but on the percentage of water content within a seed. In seed biomechanics, there are 3 classifications of seeds: recalcitrant, intermediate, and orthodox. Recalcitrant seeds are seeds that cannot tolerate desiccation or freezing and must be processed and sown quickly. Remember, this is the mango seed that I was talking about previously. Again, recalcitrant seeds are often larger, and some examples include mango, avocado, cacao, lychee. Orthodox seed can tolerate desiccation to about 15 to 20% relative humidity and store at freezing temperatures around 20 degrees Celsius. For any of you who are interested, this is actually the environment of a conventional seed bank. Orthodox seeds are typically smaller, and inadequate storage conditions are naturally longer lived. Some examples of plants that produce orthodox seeds include most annual vegetables such as peas, corn, and tomatoes. There is less known about intermediate seeds, but as their name suggests, they possess varying life spans, residing somewhere between orthodox and recalcitrant seeds. Examples include coffee, citrus, and papaya. Since we're talking about saving seeds, the rest of this presentation will be largely centered around orthodox seed, seeing as though they can be dried and stored for long periods of time. You may be wondering how you know which category of seeds your seeds fall into or belong to. And the good news is that most agricultural seeds are orthodox, and most native Hawaiian plant seeds that have been screened so far also appear to be orthodox. But there is still a lot more research that needs to be done in the field. And as a side note, just a few native species with recalcitrant seeds that cannot be saved include llama and millet. Orthodox seed can be further broken down into 2 categories: dry and wet seed. A dry-seeded crop has seeds that are enclosed in pods or husks that dry on the plant, and a wet-seeded crop has seeds that are embedded into the flesh of succulent fruits. So, for example, the picture on the screen shows a perfect depiction of a wet-seeded crop. We have tomatoes, and then a dry-seeded crop. We have beans. Dry-seeded crops and wet-seeded crops require very different processing methods from really, 2 sides of the spectrum. So to get from the field to the refrigerator often varies drastically. Therefore, we will discuss the basics of harvesting and processing dry and wet seed in turn. Let's start off with wet seeds. Because fruit encapsulates seeds in protective outer armor, wet seeds are less vulnerable to certain damaging events such as rain, but they are prone to pests. So when and how do we harvest our wet seeds? Depending on the specific crop type, fleshy fruits have different maturation times, as mentioned before. As we discussed, ripe tomatoes, peppers, and winter squashes can be harvested when they would be for eating, whereas vegetables, including cucumbers, summer squash, and eggplant, must be left on the plant much longer. In general, seeds in wet-seeded crops will continue to increase in size, quality, and viability if left on the vine or plant after fruit reaches edible maturity. So the longer you leave this fruit on the plant, the better, as long as it doesn't rot. Now, I'm gonna summarize here like the general operation of wet seed processing, and we'll talk about fermentation, we'll talk about decanting, we'll talk about rinsing, and we'll talk about soaking. So to process wet seeds, we first remove seeds from their fruit, and to do this carefully, cut open the fruit and extract seeds and any pulp surrounding them. We don't want to hurt the seeds, right? So if they have pulp that is attached to them, we'll probably just leave it on there, because there are methods later on in the processing journey that will help us remove that pulp. So next, to clean the seed from the pulp and other excess material, we'll use a combination of soaking, fermentation, decanting, and rinsing. I'll just briefly describe kind of each of these. So soaking involves placing contents of wet seeded fruit, including seeds and pulp, into a container full of water. I would advise you to allow the seeds to sit in the water until the pulp seems easier to separate from the seeds, and this should really take no longer than 8 to 12 hours. Fermentation is a processing technique for wet seeded crops that, one, makes seed cleaning easier through loosening attached pulp, two, removes germination inhibiting gel from the seed, and three, destroys specific diseases. Though experts kind of disagree on which seeds benefit from fermentation and which seeds could be harmed, the fermentation of tomato and cucumber seeds are generally recommended. Decanting is a process that utilizes a lot of water to both clean seeds and separate lightweight, less viable seed from viable, heavier seeds. And this process is pretty similar to washing your rice. So when you're washing your rice and you see chaff, or you see kind of bits and pieces of rice granules float to the top, you'll often pour those off. And that's the same for decanting, the process of decanting. You'll pour off the chaff, pulp, and non-viable seeds that float to the top, and you'll leave the heavier, viable seeds on the bottom. The final process usually involved in processing wet seed is rinsing, and this is pretty simple. It involves using a screen or a colander and some pressurized water, and you'll just kind of use that along with your hands or some sort of tool that you can move the seeds around, and that'll further separate pulp from the seed. Following these steps, clean seeds are positioned on a plate or screen to air dry thoroughly in a well-ventilated place out of direct sunlight. This is a perfect time now to pause while our wet seeds are drying and jump over to the processing procedures for dry seeds. So let's talk about dry seeds. Because dry seeds usually dry in the field on the plant, they are typically more vulnerable to weather events such as rain that could easily break through the thin capsule surrounding the seeds, potentially leading to seed germination and or molding, which you can kind of imagine is not preferable when saving seeds. So some things to look for that will indicate when dry seeds are mature and ready for harvest include the color of the seed or seed pod. So mature seeds and seed pods are no longer green and will usually change color to beige, brown, yellow, or even black, depending on the plant. We also want to feel for the dryness of the seed or the seed pod. Seeds and seed pods are mature when they reach an adequate level of dryness. So you can actually test your seed pods for dryness by maybe rolling them between your fingers to see how easily they shatter when we're talking about seed pods. You can also try bending a seed pod, and if it's leather, then, or if it's leather-like, it's usually not dry enough, but if it cracks open right away, then that's a adequate measurement for dryness. Some seeds, such as onions, can actually be cut open to check dryness. If the inside is no longer milky, and has become starchy, they are mature. And then, of course, we can also tell if seeds are mature in some ways by their ease of detachment from the plant. So this is basically how easily seeds are able to be removed from the plant or the seed pod. It's important to note that seeds do not all mature at once on the same plant or within the same planting. In fact, the span between the first ripe seed and the last ripe seed can last between 4 and 8 weeks for vegetable type crops and much longer for natives. So my advice would be to allow seeds to mature as long as possible on the plant, but harvest as quickly after maturation to avoid damage or losses from birds, insects, disease, or weather events. If time doesn't really allow for multiple seed harvests to take place, you will need to strike a balance between waiting for later maturing seed to ripen and harvesting the earlier maturing seed before they are damaged. And if bad weather is threatening, you can pick some dry seeds before they are fully mature, as most crop varieties will continue to mature off the plant. However, this is not the case for every plant, especially for native seed species. For dry seeds, again, their measure of dryness is a measure of their maturity. I'm just repeating that because it's very important. And dryness can actually help to hasten or ease the process of processing, which I will kind of go over now. But when your seed crop is dry enough, you will separate seeds from the non-seed material, including leaves, stems, and pods, as well as soil and weed seeds, using a combination of the procedures listed on the slide, so threshing, screening, and winnowing. Threshing will remove seeds from their protective outer covering. For smaller batches of seed pods, hand shelling is an effective method, of course, and if you have a lot of students who are eager to break open bean seeds, then why not? For medium batches of seeds, pods can be placed in tarps or in pillowcases or in a bucket and gently stepped on. If you have a pillowcase, you can actually just go and slam the pillowcase on the wall or on the floor. And then large seed collections can actually be sandwiched in between tarps and driven over with a vehicle. Now, I've seen a lot of seed companies or seed farms do this, but you have to be careful when utilizing this method of actually driving over your seeds, because there are different levels of hardness to a seed. And if seeds are not able to tolerate such weight, then they will crack. We're just wanting, again, to remove the seeds from their outer covering, which could be a pod, a capsule, or shake them off a seed head. And for that, you don't usually need a lot of pressure. You just need a lot of vigorous repetition. Speaking of vigorous repetition, screening is a process that utilizes that to really separate seed from non-seed material after the seed is removed from that material. So seed screening techniques vary from utilizing layers of subsequent screen sizes stacked upon each other to, again, just that simple single screen that you shake, where the larger material will stay at the top and the smaller material will fall through. And then the final process is winnowing. And winnowing basically uses an airstream to separate seeds from lighter chaff or inviable seed. When winnowing seed or non-seed material, when winnowing seed and the non-seed material is poured before an air current, and this can either be a box fan or a source of natural wind, and the heavier material will fall closer to the wind, while the lighter chaff or non-viable seed will be carried further. You would probably want to put then a bucket closer to the fan or closer to the wind to catch your viable, heavier seed. Once some or all of these procedures are complete, you should be left with a clean batch of seed ready for drying. So now we have both our wet and dry seed ready for these next steps. At this stage, when seeds are dry to the touch and clean, they're often stored in various vessels, including glass jars or envelopes that are then placed in a cabinet or in the refrigerator. While this may work in some places on the mainland, this is not an appropriate procedure to follow here in Hawaii if you wish to save seeds for long periods of time because of our high levels of humidity. This is because most seeds use moisture as one of the triggers to know when to begin the plant's growth. However, when moisture is present without the presence of other growing conditions, moisture can actually cause seeds to age faster because it triggers their metabolism. In most cases, 90% of seeds stored in unregulated conditions, so conditions that are not climate controlled, will lose their viability within 1 year time. To ensure your seeds last longer than 1 year, you have to make sure they consume their energy reserves slowly. In summary, the deeper they sleep, the slower they eat, the longer they live. Similar to animal hibernation, but again, this is called seed dormancy. What makes a seed dormant? Precisely the opposite of what makes them sprout. So when given what you want, or when given warmth, moisture, and light, a seed will sprout. So in order to ensure a seed remains dormant, you have to make sure it is placed in a cold, dry, dark environment. That forces it to really slow down its metabolism and therefore last longer. Freezers and refrigerators are the best seed storage vessels I know of. But before seeds are directly placed into these controlled environments, they must first be dried. We all know that water expands when it's frozen, right? I mean, who here hasn't seen a sealed bottle explode in the freezer, or at least expand? The cells of seeds are no different. These cells are composed largely of water and can rupture and die when the water they contain expands under freezing conditions. By drying seeds, we remove enough water to prevent this damage from occurring while leaving enough water for basic biological functions that keep the seed alive. Research has shown that there is a Goldilocks zone of relative humidity seeds need to meet before they are frozen. This range is greater than 25% and less than 35% relative humidity. When storing seed in the refrigerator, the relative humidity is a bit less precise as the temperatures are not as low. The rule of thumb for storing seeds in the refrigerator is that the temperature of humidity levels should add up to less than 100. So the temperature and the humidity levels should add up to less than 100. This means that basically if the temperature is 50 degrees Fahrenheit, then the humidity has to be less than 50% and vice versa. There are various ways to dry seeds, and today I will briefly share two of my favorites. One process of drying seeds is in a humidity-controlled refrigerator, and the other is with the use of silica gel beads. When you plan to store your seeds for a long period of time, I would suggest storing them in the freezer, whereas if you're storing them for short to medium periods of time, between 1 and 5 years, I would suggest storing them in the refrigerator. When planning to store seeds in the freezer, I would also advise using the latter method of drying with the silica gel crystals because it is a bit more precise in comparison to drying seeds in a humidity-controlled refrigerator. So I'll talk a little bit about this process of drying seed and then we'll move on to the next. So self-defrosting or humidity-controlled refrigerators can be utilized as drying cabinets for seed. In general, these refrigerators keep the air dry at adequate levels for seed drying. However, units may vary depending on their use. So if you have a lot of produce or liquids that are stored inside the refrigerator, obviously the humidity is going to go up. This can be controlled, however. To prepare seeds for drying, spread them in a thin layer in a shallow, wide-mouthed container. Choose an airtight container, but with an accompanying lid, but don't put the lid on quite yet. You will store this container with seeds in the refrigerator for a month. And after a month, you will open the refrigerator door, quickly put the lid on the container, and then remove the container and place it in a dark place. Once the container has come to room temperature, you can open and pack the seeds for storage. Now, the reason we do this, and I'll talk a little bit about this later, where we put a lid on the container before we take it out of the refrigerator is to prevent condensation, because if you just took your seeds out, then it'd really be pointless after leaving them in the refrigerator for a month. You took them out, they would reabsorb all that moisture and condensation. So the next method of drying involves a few more materials that are listed here on the slide. I will share a summarized version of instructions for constructing your own seed drying container. First, you'll have to find an airtight container in proportion to the amount of seeds that you have to dry. The container can be see-through or solid in coloration. And if I have a small batch of seed, I like to use mason jars because, you know, they come in various sizes and you can also see through them. And this will allow you to see the change in coloration of your dry card. Second, weigh or estimate a ratio of seeds to beads or beads to seeds. They should be about equal. You don't want to have a lot more beads to seeds because if seeds dry down too fast, this could also damage their cellular tissue. Similar enough if they are, put in the freezer. So after you weigh out your ratio of beads to seeds, you'll place a layer of silica beads in the bottom of your drying container. And once the beads are spread out, you will place a divider. This can be a paper towel as seen in the screen. This can also be a coffee filter or even a cupcake kind of sleeve. I've used that before. Next, you'll place your seeds that are going to be preferably in a shallow, wide mouth vessel into your drying container. And you will quickly close your container. And then I would advise everyone to store this container in a cool, dark place until your seeds are dry. Now, depending on seed size and density of the seed lot, this can take anywhere from a few hours to a few days, and sometimes a little bit more than a week. But I would advise you again to just periodically check on your seeds and make sure that they are drying. To do this, you'll place your UC Davis dry card in the drying container and you'll let it sit for about 60 minutes. The dry card will indicate the relative humidity of the atmosphere, which is also the humidity of the seeds in the container. And once they have reached the Goldilocks zone between 25 to 30% relative humidity, you will be able to store your seed. This is when the dry card turns very bright blue. After drying, all seeds should be stored in airtight packaging or containers in the refrigerator or freezer. I like to use food grade vacuum sealing packets, but sturdy Ziploc bags or glass jars will work just as well. Again, seeds in good condition and stored properly will last at least 1 year. And depending on the plant, many will last 2 to 5 to 10 years. But again, there is variation in that. Storing seeds in the right conditions will cause them to age slower than in their natural environment. However, all seeds still age while in storage, meaning that all seeds will sadly but truly lose viability and die eventually, unless they are planted. But the viability of a collection of seeds typically follows a pattern called sigmoid or sigmoid curve. Initially, there is usually a long period of high viability with only a very gradual loss, followed by a steep drop in viability. So to follow this curve and ensure your seeds do not lose viability in storage, especially if you're storing them for longer than a year, germination testing is a necessary step. Before we explore the basics of germination testing, it's important to note again that when removing seeds from either the refrigerator or the freezer and transferring them to room temperature to either plant or test their germination status, you have to prevent condensation from occurring. And you can do this by allowing them to reach room temperature before opening the container. For seeds that sprout within 2 weeks, germination tests can be completed in a petri dish or with a paper towel and Ziploc bag. But for seeds that take a bit longer, such as many of our natives, germination tests will likely need to take place in pots filled with soil. Once seeds have been given an appropriate time to germinate, count how many seeds have sprouted, divide that number of seeds by the total number of seeds in the test, and then multiply that by 100. As you can see, the formula is listed there on the slide. If the percentage of seeds germinated seems high enough compared to when first accessioned into storage, the specific seed lot can remain in storage. But if the percentage of seed has drastically decreased, they will need a quick exit strategy, because when it comes down to it, the best way to preserve the living legacy of a seed is when those seeds are planted and growing in the soil of this earth. We can't talk about seed saving without talking about seed sharing. When you save seeds, you're joining a cycle of reciprocity in harmony with the natural path of seed, receiving, planting, growing, and giving. When you share a seed, you share a thread in the web of life, strengthening bonds of connection throughout every dimension of the earth. I believe that in order to transform the present day reality of our seed system, we need to cultivate awareness around the true value of seed, no longer as a commodity, but as a currency. And that's where seed sharing comes into play. There is a brief list of some of my favorite seed teaching tools. You will all be receiving this slide deck as a resource. So if you haven't already, I encourage you all to check them out. It's been a pretty long time of talking, of listening, and I hope you all have been able to understand at least a portion of what I've tried to share. But for now, I want to ask you all to take a second. Take a deep cleansing breath. Breathe in, close your eyes, and imagine all of the lessons that seeds provide. Lessons of patience, lessons of strength, persistence, and resilience, lessons of giving and receiving, lessons in every dimension of life, for seeds are the very foundation of life itself. By participating in life systems that sustain our earth, awareness grows through experience to spark interest, prompt questions, and cultivate creativity. This encourages the evolution of self-expression, self-exploration, through the bridging of passion and knowledge, which in fact is the definition of a leader, one who follows their heart with an open mind. So I want to thank you all for today being a leader. in this movement of seed and seed sharing, because you are now, I hope, going to take these seeds, plant them, and share them with the minds and hearts of your